drink lots of water. So it drinks water, right? But it has to pump out salt from its gills. So it pumps salt out the gills. Salt out gills. It loses water to the environment, so it loses water across the gills. And it produces just a little concentrated urine. So what this means is that fish that are anadromous actually have to shift their physiology almost completely to the opposite extreme. So they start out as freshwater, they go out into the ocean. Exactly the opposite problem is that they're losing water instead of gaining it. And then they have to use energy to get rid of the salt. And so you could almost say that a marine fish is desalinizing. So desalination of water, right? So it's drinking lots of salt water, but it's getting rid of the salt. So it has to use lots of energy. So this takes energy. The opposite with the freshwater fish, they actually actively transport ions in. So they take up ions. So they take up salt. And this takes energy. And so this is why osmoregulation is energetically expensive in both environments. So the same thing happens when we talk about marine birds and marine turtles. So when we talk about marine birds, they actually have a salt gland here. And what this salt gland does is that blood comes into the gland, and what happens is that they secrete the salt out through these glands, and it comes out through an opening on their bill. So what this does is removes salt from the blood. You know, actually reptiles have this too. So like marine turtles. So it removes salt from the blood. Right? And it comes out openings in near their nostrils, or it comes out their nostrils. So it just comes out their nostrils. Okay. Because if you think about marine birds, they're going to be taking a lot of salt water, so they need a way to get rid of the salt. And so they have special glands that do that. And the salt glands on turtles, marine turtles specifically, actually makes it look like they're crying, right? And so this is actually just a salt deposit. So they're not actually crying, they're just getting rid of the extra salt that they have taken in. So it's just a mechanism, yeah.
So what about birds that like they migrate to like marine areas? Like when they're breeding, are they like does that part of their nostrils like become like dormant? Or
I would imagine that if there's not very much salt in their blood and it goes to that salt gland, then nothing will happen. But if there is a lot of salt in their blood, when it goes to that gland, then the salt will be removed. But I would imagine that the gland stays active. It's just that it probably only works if there's extra salt, because it's kind of like, it's almost like a dialysis machine that takes out the salt from the blood. Yeah. Okay. So let's talk about the different types of waste products. So one thing about our urinary system and our excretory system is that it also not only gets rid of extra water, but it also gets rid of nitrogenous wastes. And so these are produced when we break down proteins and DNA. So if you have a meal high in protein, DNA is in everything, it's in all cells, right? But if you have if you're eating meat, for example, protein and DNA would be present. And when you break that down, you get a waste product. And it is nitrogen based, right? So nitrogen based.
What happens if you're eating synthetic proteins like tofu? Or
it's the same thing. Tofu is actually soybean, so every plant.
I don't mean synthetic,
yeah, plants. Yeah. So protein, so any protein is going to cause nitrogenous waste to be produced. So sometimes if you have a diet high in protein, it actually puts a little bit more stress on your kidneys, because kidneys are what remove the nitrogenous waste from the blood. So when we look at the nitrogenous waste, we can talk about, so I'm gonna label my waste, but I'm gonna create a table here. So I'm gonna put toxicity in one of them. I'm also gonna put the amount of water needed to get rid of. So I'll put needed to excrete. And then I'm gonna put energy needed to produce. Okay. So the first type of nitrogenous waste is very simple in structure, and it's just ammonia. And if you've ever used ammonia for cleaning, you know that ammonia is very toxic, right? So we would say that it has a high toxicity level. We also would need to dilute it extensively. So we would need lots of water. I should use a different color. We need lots of water. Okay. And then it would require little energy to produce. So there's a trade-off. Ammonia is very toxic. It requires lots of water, but it doesn't require a lot of energy. So animals that have access to a lot of water use ammonia. So if you think about freshwater fish, for example, even tadpoles of amphibians that live in freshwater. So freshwater organisms like fish and amphibians would use ammonia as their nitrogenous waste. Okay. So at the other extreme, we have uric acid. Okay. It has a low toxicity. We need little water, but it takes lots of energy to produce. So uric acid is actually a solid. So this is not what we use as a nitrogenous waste. So what type of organisms produce uric acid, do you think? It's white. It's a white paste. So we see feces that have a white paste in it, and it is birds. Okay. So birds and reptiles. So this would be like freshwater fish, birds, reptiles, but also insects use uric acid. Insects that, specifically the terrestrial ones, right? Grasshoppers, for example. So this has an advantage because it doesn't require a lot of water. So this would be an evolution towards a terrestrial lifestyle where you don't have access to very much water. And you can just get rid of your uric acid with your digested waste, because both reptiles and birds are reptiles now, but they have just one opening. They have a cloaca, and so their feces and their uric acid all come out together, and that's what you're seeing on your windows, right? White and brown or white and blue, right? They're eating berries. That's the uric acid. Okay. So we use urea. And this is just moderate. Moderate. Okay, so we don't have to dilute it as much, so that means that we don't have to, get rid of as much water when we urinate. If we use ammonia, we would have to like dilute it within a lot of water, and we have to urinate it out with lots of water. Urea can also be secreted in our sweat glands. So sometimes when we sweat, we're also getting rid of urea. Okay.
Is it high stress on the liver as well, with high protein diet, or just on the kidneys essentially? Because you said it was kind of high stress on the kidneys.
I don't think it's high stress on the liver. Yeah, just the kidneys. Only if the meat has toxins in it. Yeah, would it be liver? Okay, so in your book, there's a diagram that shows the structure. Ammonia is just NH3. This is urea, a little bit more complicated. This is uric acid, right? So urea, ammonia. So there's just trade-offs to the toxicity as well as the amount of water and the energy required to produce it. Okay, so why don't we take a 5-minute break, and then we'll come back and we'll talk about the different types of excretory systems that we have in our body.
You want our extra credit review right off hand?
I can look it up. So it should be in the Canvas.
What do we have to do?
I have the handout, but I don't know.
Michelle?
I'll show you. I'm going to look it up. One more. Oops. Okay, so on the home page, you come down here and it's extra credit assignment right there. And it is due, oh, I haven't decided when it's due. I don't think it's published either. Oh, it is published.
The max on that, 15 points.
Generally.
It says here on this handout 5.
I know, sometimes I give people extra if they do it outstanding. Generally, it's 15. Okay, so your thing is due the 12th, your formal project. So we'll make this one due the 18th, just as long as that does not interfere with your studying. You don't want to spend a lot of time on it.
For our formal lab report?
You don't, yeah, so you don't want to spend, no, you don't want to spend a lot of time doing your extra credit instead of studying for your midterm. So you could actually do your extra credit now.
I guess you said our formal lab report would write us through the 12th.
Yes.
So if we choose to do like that planaria experiment for our formal lab report, we're not required to do a informal lab report on that same one.
Yes. Just put it right in your notebook. So if you're okay, so if you're already starting to write up that plenary regeneration, right? And it's the formal lab report, put a copy of it in your lab notebook so you can get credit for it. Plus you'll get double credit for it, right? Because you don't want to have, you want the lab write up, so you have to have just put it right in there. Yeah.
So instead, rather than doing the lab informal.
Just put the
Yeah.
formal one right in there and I'll just grade it. Yeah. So you already did like the thermoregulation, right? And so if you're going to do thermoregulation, that's already in your lab notebook as an informal. Right.
But add a few things to it. And
Right.
make it fun.
Yeah. Oh, I'll be right back. Oops.
You said that was through the 18th?
Yes.
Who's really getting like 3 days to start? Everything will enter. Alrighty. It's good to have time.
Okay. So when we talk about excretory systems, we see an interesting kind of development over the different animal groups. So the phylum Platyhelminthes would be the flatworms. So that would include planaria, but it would also include tapeworms and flukes, so those endoparasites. So when we look at the Platyhelminthes, they have what we call protonephridia. And these protonephridia have these interesting structures called flame cells. And the flame cells have cilia that create a current. And this current draws the fluid into the excretory system. So a current that draws fluid into the tubules. Okay. So if we look at a structure of these protonephridia, okay, this is the excretory system of your planaria. So this is not the digestive system, although it kind of looks like it. So they're just showing the excretory system. So how do you think planaria get their oxygen into their body? Through the surface of their skin, right? So oxygen moves in and carbon dioxide moves out. But with that oxygen moving in, water also moves in. So water would be constantly moving into this organism. And so one way to get rid of that, as well as to get rid of their nitrogenous waste, is to filter that fluid and then get it back out. And so you can see here that there's a cell that has these cilia and they whip around, and then they create a current which actually sucks stuff up into the tubules. And then they have these openings in the body wall, and so they just kind of pump the fluid back out. And with that fluid goes the nitrogenous waste. So the nitrogenous waste exits that as well. Okay. So those are the protonephridia. So then we could talk about what are the nephridia. And these are actually in the phylum Annelida. So what organism did we dissect that is in the phylum Annelida? Earthworm, right? So the earthworm have nephridia. And so these are in each segment. So they actually urinate out of each segment. So remember, one of the advantages of having earthworms in your soil is they eat dirt, which has bacteria and fungi in it, and then they excrete nitrogenous waste. So the waste comes out, and then that nitrogen can actually be used by plants to grow and to develop. So if we look at the nephridia, they look like this. So this is in blue. Remember that they have a closed circulatory system. So the capillaries are in red. So the nephridia are in blue. And they just draw in the coelomic fluid. And then the good stuff gets taken back up by the blood. And then the nitrogenous waste comes out through these openings in each segment. So they just are urinating out of each segment. Okay. Now there is another type of excretory system in the insects that is called the, so if we look at insects, this is phylum Arthropoda. They have branches off their digestive system that have kind of a cool name. They're named after a person, which they're called the Malpighian Tubules. So these branch off of digestive system, and they just filter the coelomic fluid, so the fluid that is in the body cavity. So if an insect takes in extra water and it also eats protein, it's going to produce nitrogenous waste. If it takes in extra salt, you can also get rid of the salts here. And so it just passes it into the digestive tract, and then it comes out with the feces. And so like birds and reptiles, they just have one opening to their excretory system and their digestive system. So the excretory waste and the digestive waste come out together. So if you've ever had a grasshopper poop on your hand, that's what that was. Okay. So we do not have tubules located out throughout our body. All of our tubules are in the kidney. So they're concentrated in the kidney, and the tubules are called nephrons. These are the tubules in the kidney. So blood travels to the kidney, and then it is filtered. So if we look at the function of the kidney, so if we look at function, we have the filtration of blood. We also have a filtrate that we then have to modify. So we have reabsorption of substances from the filtrate. Okay. So here we would go from the filtrate that has been produced, and it's in that tubule, the nephron, and it's going back to the blood. So let's think about like glucose. So if I filter glucose out of my blood, I don't want sugar in my urine. In fact, that's a sign of what disease? Diabetes. So if you have sugar in your urine, that's a bad thing. It means your blood glucose is too high and you can't actually reabsorb all the glucose. So I want to reabsorb the glucose and take it out. I also don't want protein in my urine. So my amino acids want to come back out too, right? So I want to pick that stuff back up, okay? And then we have secretion. And I'll put of substances into the filtrate. Okay, so in this case, it goes from the blood to the filtrate. And one big thing that we get rid of in this way are hydrogen ions. Okay? So if we have too much hydrogen ions in our blood, that means our blood is too acidic, right? So as it's filtered by the kidney will sense that, and then the blood will actually cause the kidney to take out hydrogen ions, and then your urine will be more acidic. So the acidity of your urine actually varies quite tremendously on what it is like you're eating, right? Because different foods can influence the acidity. It can also be influenced by your respiratory system, because that also influences the acidity of the blood. Okay, so that's the function. So if we look at the types of nephrons, the main type of nephron that most animals have are what are called cortical nephrons. So if we look at the structure of my kidney, the cortex of any organ generally is the outside. So if I look at my cortex of my kidney, it would be the outside. So this is the cortex. The inside is called the medulla. So the outside is called the cortex, the inside is called the medulla. And so the cortical nephrons are only located in the cortex. Okay? So in us, about 85% of our nephrons are cortical in humans. This will make sense because when we talk about the function of the nephrons, what's really important to us are the nephrons called, so there's two different types, which are called the Juxta medullary nephrons. So juxta means next to the medulla. So these extend into the medulla. And although they only make up 15% of the nephrons that we have in our body, in our kidneys, and there's thousands of nephrons, these are the nephrons that are capable of producing highly concentrated urine. So these produce highly concentrated urine, which means that they are responsible for conserving water. So they conserve our water. So that if we go without water, we should start to produce very concentrated, just a little bit of urine, over a given period of time. When we become dehydrated, we're not going to urinate as much. And it's because of those juxtamedullary nephrons. So let's look at what these nephrons look like. So here's a diagram in your book that shows the blood actually coming in. This is the renal artery carrying blood in, and they say like 2/3 it 2/3 of your blood? Maybe that seems like high. I can't remember what percentage of your blood actually goes to the kidney every time. But anyway, so a lot of blood comes into the kidney to be filtered. And then this is carrying the blood back out. Okay. So these are the parts of the nephron, and I'm actually going to draw it because I think that will help us understand it. Okay, so we have a series of capillaries which are called the glomerulus. So this is my capillary bed where filtration occurs. So remember, capillaries are leaky. So we want blood to come in under high enough pressure that a filtrate is going to be formed. So actually, it's really, blood pressure is really important to the kidney because if you have excessively high blood pressure, it can damage the kidney because it can blow out the glomerulus, which is the filtration membrane. Okay. So if I drew my blood vessels, like here, and they come into the glomerulus, like this, and then they come back out, okay? So those are my capillaries. Now, surrounding the glomerulus is the tubule that is going to collect the filtrates. Okay. And this actually has a Bowman's capsule. Could be the renal capsule, but the Bowman's capsule. So that collects the filtrate. So hopefully red blood cells do not get into the urine. So we don't want red blood cells. Hopefully we don't have white blood cells. That suggests we have a urinary tract infection, right? Bacteria also. So only things that are soluble in the water come into the tubule, okay? And then this tubule kind of winds around. So I'm just gonna put some little, okay, little meanderings in it. And then we're going to look at one of those Juxta medullary nephrons that extends down into the medulla. So this tubule extends down into the medulla. Okay. So this is just one of thousands of nephrons that are all aggregated into your kidney. Okay. So this is medulla. This is the cortex. Okay. So this first set of tubules, first part of the tubule, is referred to as the proximal because it's nearest, convoluted because of its shape, tubule. PCT, it's abbreviated as. This, because it's further away from the Bowman's capsule, is the distal convoluted tubule. Okay. Does anybody know, has anybody studied this before? What is this called? It's kind of famous. What is that thing that extends down? It's the loop of, nobody studied the kidney before? It's famous, loop of Henle. Okay. So the thing about this is that different things happen in different parts of this nephron. So this nephron here, we have reabsorption. So this would be like glucose, amino acids, salt. Things get taken out of the filtrate. So that is called reabsorption because it's moving from the filtrates back to the blood. And there's capillaries that are surrounding that. Okay. Here in the distal convoluted tubule, we have secretion. So let's just put a hydrogen ion there. So that means that we're secreting hydrogen ions into the filtration. And then something interesting happens in the loop of Henle. So here, water is drawn out all along the length of this what they call the descending limb. So I'll put the descending limb. Because the filtrate is moving in this direction. So it's moving down, right? It's coming here, it's moving down and around, okay? So then this makes this the ascending limb. Now water moves out passively. So this is an example of osmosis, right? So it is picked up by the circulatory system. So there's capillaries surrounding my loop of Henle, and it's picking it up. This does not take energy, so no energy. It's not being pumped. So we're actually using the way that this is set up to reabsorb water, because as we move down into the medulla, the salt concentration is higher down deep in the medulla than it is up. And it just sucks the water right out of the loop of Henle. So if there is a cortical nephron, it doesn't extend down into the loop of Henle as far, into the medulla as far, and so water is not reabsorbed by the cortical nephrons, okay? So on this side, on the ascending limb, we have salts moving out. Right? And so this is actually takes energy. So it's not passive, right? So this is what sets up the solute concentrations in the medulla that draws the water out. Okay. So would you think a beaver or a desert rodent would have a larger or a longer loop of Henle? Would a beaver or a desert rodent have a longer loop of Henle? Which one? Desert rodents, right? Because the longer the loop of Henle, the more water is going to be reabsorbed across the surface, okay? So if we look at a diagram that shows this, oops, to go back to my topic, sorry. Oops. So in this diagram, we are actually kind of like the rabbit, okay? So this right here just shows the difference between the cortex and the medulla, the dotted line, right? So rabbits, we're kind of like rabbits. We have these loops of Henle that extend a little bit down, but a kangaroo rat is able to conserve so much water that it actually doesn't even ever have to drink water. So if we looked at the water balance in us compared to the desert rodents, like a kangaroo rat, and we have these like down in the southern part of the state, notice that they do not drink water at all, right? We have to drink quite a bit of water. And the reason why we have to drink quite a bit of water is we're not able to get as much water from breaking down our food as they do. So they just are very good at getting water from their metabolism, right? And then they actually lose more water through evaporation because they live in the desert than your typical human. But notice how they produce very little urine, and it is very highly concentrated. So the only other thing that we need to draw on that diagram that I put up there is what is called the collecting duct. Okay, so I'm going to extend this out a little bit. So this would all go to and I kind of wrote all over it. So this would be my collecting duct. So each collecting duct has multiple nephrons attached to it. All of the filtrate is then going to be collected, and it's just going to drip out of the kidney, and it's going to go out through the ureter to the bladder where it is stored, and then it goes from the bladder out to the urethra. Okay. So that collecting duct, we're going to talk about the significance of that collecting duct and how we can produce more concentrated urine through hormonal mechanisms, including the antidiuretic hormone, or ADH. Okay. So we're going to stop there for today, and we'll finish up on osmoregulation, and then we're going to start on the nervous system. No, the collecting duct is still in the kidney, but it's going to collect all the filtrate from all the nephrons and then take that filtrate right to the bladder. So it just kind of drips into the bladder from the kidney.